So students, the factors which affect the enzyme's activity are different. So I have only drawn the graphical representation to represent them. For example, when we talk about the, see, brackets always mean the concentration. E means enzyme, P means product, S means the substrate. So as the enzyme concentration increases, there will be always an increase in the speed, reaction velocity. But remember, a time will come, it will become a straight line or a constant value. The reason, there are other factors also involved. You cannot simply keep on adding enzyme. The other factors also have to be increased, then only the enzyme activity is going to increase further. The second is the product. As the concentration of product increases, enzymes activity will slow down, because we all know enzymes are have greater affinity for the substrates to join to. As the product concentration increase, the reactivity is going to come down, so the speed will go down. If you remove the product, speed will increase. If you add the substrate, speed will increase. Now the next one is if the concentration of the substrate is increased, you will find there will be a increase followed by again a constant line. Now this is a topic explained in a great detail in NCERT. Now the main thing is, but the maximum velocity, see, for a reaction to go on with a particular substrate and a particular enzyme, the increase will go on like this. You increase the substrate amount, it will increase further, it will increase further, and finally it will become a constant value. That value is the maximum velocity for a reaction, and that concentration is this much. Now to understand this concept better, Michaelis and Menton, one Canadian, one Australian, two scientists have given a concept, and this is called as the Km value. So what is Km value? M stands for Michaelis and Menton, and K is the constant. And the Km value is what? It is the value, it is the amount of the concentration of the substrate. So please pay attention, this was this concentration required for maximum speed. So what is Km value? It's the amount, it is the concentration of the substrate at which the Vm value, at which the speed of the reaction is halfway of the Vmax. So at the point when the Vmax become half of the Vmax, the velocity becomes half of this. That is the concentration of this, that concentration of the substrate is called as Km. I'm repeating again, Km is the substrate concentration at which the speed of the reaction is half of the Vmax. And generally for enzymes, this Km value is 10 raised to the power minus 1 to 10 raised to the power minus 6 m. And remember, if the Km value comes down, then the enzyme has more substrate affinity, and the Km value differ from enzyme to enzyme. So every enzyme will have a different Km value. Plus, we all know some enzymes are able to involve with more than one reaction or more than one substrate. So every enzyme will have different Km value for different substrate also. Now the next is some enzymes need an optimum hydration also. Enzymes need an optimum hydration also. For example, we all know water is the universal solvent. So we know every reaction in the living system is occurring inside the water. So how much concentration or how much hydration is there is very important for different enzymes. So enzymes have an optimum hydration required to function properly. Next is the activator. We have already learned the activator topic in the proenzyme, that some enzymes are proenzyme, they need to be activated. So if the activation conditions are missing, they will not become active. So example, chlorine ion or chloride ion is required for the salivary amylase to function properly. So without chloride ions, the salivary amylase of saliva will not function. So proenzyme is made into active enzyme with the help of the chloride ion in this case. So chloride ion is the activator here. Next is temperature. NCRT has drawn this diagram in a very easy manner like this, the way this one is drawn, but reality is this way. So same way we have pH also. So enzymes activity increases from minimum, it goes to the maximum. So now remember, the speed starts from here. As the temperature is minimum and it goes to the maximum, so you will find maximum enzymes have their efficiency close to 25 to 35 maximum, and then it will quickly start coming down. So it won't be a bell-shaped structure, it will be like this. Reason enzyme starts quickly turning into denatured one, so their speed will finish. So no more enzyme activity is possible. So it's a sharp decline, it's not a smooth one, sharp one. Reason denaturation will take place. So if it is very less temperature, enzymes are inactive because of freezing temperature. If the temperature cross the 40-degree mark or 45-degree mark, enzyme will no more function because they are denatured. Optimum is what? Basically 35 for maximum. The next is the pH. So pH value, this will be a bell-shaped jar. It will increase. It won't function beyond a particular pH. It will function in a range of pH, and in that range the maximum speed will be somewhere in the center. So optimum speed. Remember, most intracellular enzymes function near, I have already told you this point, most enzymes which are intracellular enzymes, they function near the neutral pH. Now the next important topic is inhibition. So how inhibition of enzymes activity take place? First is, if the protein is denatured, enzyme will not function, because how proteins can undergo denaturation? High energy radiation, strong organic compounds, salts of the heavy metals, these things can cause denaturation or the bonds to be broken. Now inhibition of enzymes can be temporary or it can be permanent also. So